Please make sure your cell phones are silent so we don't disturb our speaker, who is tonight Cameron Jones, who is a master's student in the Vargas Lab here at Cal Poly Humboldt and has been studying wallflowers. So tell us whether there's more than just Menzies wallflower out there. Cameron, I'm gonna let him introduce himself a little bit more because he's got a pretty interesting job now too.
Thank you for that introduction. So before we dive in, I want to know, how's my volume? Can you hear me back there? Pretty loud and clear? All right, that is spectacular. Today we're all here to figure out what is going on with Menzies wallflower. We want to remove that blurry, we want to address the species boundary, which overlaps with adjacent allies. Sorry, this is my first time. I haven't taught in like a semester, so I'm a little rusty when it comes to public speaking. So give me some grace. We'll go through it. I also want to mention I have a speech impediment, so sometimes I might stutter or I might get stuck on a word. We will work through that throughout this talk. Just give me a second, and we'll keep going forward. All right. So now back to the subject at hand. Today I'm here to discuss the species boundaries in the Menzies wallflower. This is a wallflower found here locally in Humboldt Bay. It is also found in Mendocino County, as well as in Monterey, and potentially in Donner County, underneath what is colloquially called the Tolowa wallflower. But we'll discuss that further in a second. But how did we even get here? There's been numerous studies throughout the last 100 or so years trying to assess Erysimum as a whole, especially in North America. Rosenbach in 1903 was doing a key for the Erysimum found in North America as well as in North Mexico. While writing that key, he had a statement. So the statement was, Erysimum cancinum, aka the Helen wallflower, appears to be an intermediate between Erysimum menziesii, the Menzies wallflower, and Erysimum capitaneum, the Western wallflower. I guarantee most of you have seen that Western wallflower. It's the orange wallflower you'll find usually at higher elevations. And he said this might be a hybrid. Essentially, the morphology was kind of in between what you see with capitaneum as well as menziesii. So there was some blurry species boundaries right there. And then we jump forward to 1987, about 80 years prior to that previous paper, or after, where Robert A. Price wrote his dissertation about the Erysimum capitaneum alliance. In his dissertation, he noted that Erysimum cancinum, aka the Western wallflower, the Helen wallflower, and Erysimum cancinum, Menzies wallflower, may be sister to one another, which just means they're closely related to one another, and potentially descendants of capitaneum. But at the time, all they had was looking at chromosomes and a morphological assessment. There was not the means to understand that relationship further at the time. And then in 2003, there was a study about the genetic affinities of a wallflower or Erysimum population in Donner County, specifically near Lake Earl area. That's where they're addressing the Tolowa wallflower. They wanted to see where that population lined up genetically versus morphologically, because that population has always kind of been vague. And in that study, they said it was more likely, it was closer to the population adjacent to it, which was a continued population at Weathers Island. But they also noted that the taxonomic position of the Lake Earl Dunes population might be better understood if we did a more broad sampling plan. In that study, they sampled one population per species, so there was less context to what was going on at that locality. And then lastly, in 2014, there was a taxonomic analysis of the coastal Erysimum menziesii. This was done by Lena Vorpic. This was to address the previous research where there wasn't enough genetic information, so she surveyed more localities throughout California. And sadly, at the time, the technique that was used just wasn't adequate enough to differentiate these populations to the level we needed to get some clarity. And then we jump to 2022 in February, where Senator Trebek and Katrina Henderson were giving a talk about the Silophysalia found in the Tolowa Dunes. During this talk, they noted there is also some interesting taxa within that region. One of those interesting taxa was the Tolowa wallflower, also known as Erysimum cancinum. And in this talk, they mentioned that perhaps we should reevaluate it, and perhaps it more so aligns with this rank you see right here, CPS rank 1B.1, maybe it should be considered rare and endangered and critically threatened. At this time, this was assessed as Consinum. Consinum is considered vulnerable, but it doesn't have the same rank that you see with menziesii, unfortunately. And at the end of this talk, they mentioned hopefully they get a genetic assessment to occur in the next couple of years. And thankfully, several months later, my advisor, Dr. Oscar Vargas, gave a talk discussing his research with the California Forest Province, as well as the neurotropics, discussing how we're trying to understand diversity. And how we do this is using phylogenetics. Sorry, wrong arrow. Phylogenetics being the study of evolutionary relationships between organisms. Imagine this as a family tree, but usually for a certain system. So if you want to understand what's going on with Erysimum, perhaps this would be a great metric. And Sandra noticed that and reached out to my advisor, Dr. Oscar Vargas. And Dr. Oscar Vargas mentioned to me, hey, Cameron, there's this cool wallflower in Dark County, which with a kind of vague identification. We didn't know if Tolowa wallflower was Mencinum or if it was menziesii. So we need some clarification through a genetic means. And when Oscar mentioned it to me, I was ecstatic. For those who do not know, I'm from Dundalk County. That is my hometown. That is where I was born and raised. I'm also Tolowa. So this immediately felt like a cool homage to my home community. At the time when Oscar proposed this to me, I was still trying to figure out what to study exactly. I still wanted to address an evolutionary question, but now I had a medium to study that through Erysimum. And from there, I had to kind of figure out my angle for my thesis project, was just to more so address address diversity occurring within a California Forest province, an area that our lab likes to try to add to or get more context to what is going on. This area is renowned for being a major biodiversity hotspot. There's over 6,143 total vascular plants within this region. 2,612 of these are endemic to the region, meaning they're only found within this defined area. And 1,842 being considered rare. Previous studies have aimed to understand this origin diversity, but they more so focus on evolutionary processes affecting above the species level, as well as going more at the family or the genera level. Leaving there to be little known about what is driving diversity at the species and the population level, which could be contributing factor to what is going on with Menzies Wallflower as a whole. And there's some Menzies Wallflower is just the perfect medium to understand what is going on within the California State Province and why it is so diverse. It is a coastal dune endemic, meaning it's only found within the coastal dunes. It is considered rare and endangered, being 1B.1 through CNPS, and being considered federally endangered. It's also even globally endangered. There's been 3 previous recognized subspecies. Subspecies ericensis, which is the Humboldt Bay wallflower. That's what we have here locally. We have subspecies Menzies wallflower. Then we have subspecies Yadon's wallflower. And potentially the Tolowa wallflower. Perhaps maybe the Tolowa wallflower should be more closely aligned with what is going on with Erysimum menziesii. Now, on this map to the right here, you can see the most northern extent, which is kind of a turquoise color. That is where we have the Tolowa wallflower. Moving south of that point, we then see we have the Humboldt Bay population. That is Erysimum menziesii subspecies ericensis. Moving south of that locality, we then have subspecies menziesii being found, as well as subspecies menziesii being found in Monterey. Then lastly, Yadon is also found in Monterey. Sorry, I keep hitting the wrong arrow, my bad. It's also a member of this alliance known as the Erysimum Capitam Alliance. This alliance is based on the premise that all of the Erysimum found in the Pacific Northwest or in California, or more so in California, share the same unique chromosome count, which is 2n equals 36. Typically in Erysimum, it ranges from 2n equals 12 to 32. This suggests that all the Erysimum in this alliance is closely related to Erysimum capitam, that western wallflower. Previous morphological studies hypothesized that Erysimum Menzies wallflower, and Erysimum cancinum are derived from Erysimum capitam, that western wallflower. Also, Erysimum cancinum and Erysimum menziesii overlap morphologically and geographically, suggesting that that these two might be sister to one another, meaning they're closely related to one another. Now, why is this named after the Western wallflower? That is the most broadly disputed wallflower that we have. It is, like the name says, it is found throughout most of the Western United States. Essentially west of the Mississippi and a little bit east of Mississippi, you can find it. Usually it's in more mountain regions. You kind of notice where there's some topography, you kind of see more occurrences pop up. It also expands into Mexico and even into Canada, even into Alaska. It has a broad range. And then we move into California, we can still see those occurrences pop up more so in higher elevations. But as we get kind of closer to the Northern California coast, we start to see occurrences dissipate. And then another competitor jumps into the battle here. We then see Erysimum cancinum pop up, the Helen wallflower. The Helen wallflower is to be found on coastal prairies or cliffs. You can see in Donner County, kind of in the Lost Coast Range, Mendocino County, as well as in Point Reyes area. And then typically adjacent to those populations, we'll find Erysimum menziesii, Menzies Wallflower. Often these two kind of flip-flop on their localities, like in Donner County, we have Cancinum, Humboldt, Menziesii, we may have Cancinum again, and so on and so forth. So it's kind of an interesting pattern, and it even overlaps in areas such as Mendocino County. So these two overlap geographically, but what's going on morphologically? So today we have a wide range of individuals in here. Some of you are seasoned botanists. Some people, you have the green blur still, as in we look at plants, everything kind of like smudges together, right? It's hard to differentiate one species to the next. So these two have confused botanists in the past. I think we all can generally say the one on the left looks larger than the one on the right, right? We also see they're both yellow wallflowers. So there is some differences between these two. We can see them is definitely larger than menziesii, but there's really only three categories of traits that really separate these two, according to the Jepson. One being fruit angle to the stem. So for those who are not super savvy with botanical terms, you see the yellow flowers below there, you'll see these green appendages kind of going outward, protruding. Those are the fruiting bodies found in Erysimum, known as siliques. They are long and sleek. And depending on the angle that the fruits are pointing, that can depict if they're cancinum, which should be more at a 45-degree angle, or menziesii, being more perpendicular to the body as a whole, or at a 90-degree angle. The stem length in cancinum is almost twice the length of what you see in menziesii, so there's a staggering difference in size. And the habitat is different for both, usually. The habitat for cancinum typically is coastal cliffs, sometimes coastal prairies, and with menziesii, it's more so in coastal dunes on a sandy substrate. So there is some ways to differentiate these two. But what happens when cancinum is found in a sandy environment? So for those who could easily tell the last two apart, at least to me, now the line gets blurry, right? There's a green blur with most plants. I would argue now there's a yellow blur with the wallflowers. Yeah, and don't worry, this has mystified the previous two botanists who have assessed these populations. And it has mystified me to a point where when I was setting up my proposal meeting for my thesis, I accidentally swapped the photo of menziesii for a photo of the tall wallflower. I didn't realize that until after the talk happened. I'm sorry, Oscar. But I also fooled my whole committee, so we're all into this together now. So yeah, so now we have a clear issue. It is hard to differentiate these species, especially when cancinum infringes on what has been menziesii's habitat. So this leads to my objectives of my project. The first objective is well, we need to clarify that species boundaries. We need to figure out what separates Erysimum menziesii from cancinum. To do this, we'll use a phylogenetic analysis. It's basically like a family tree, or more so a tree of life, to understand how closely or distantly related these groupings are to one another. We also want to know what makes Erysimum menziesii distinct. Is this a certain morphological feature? Is it a soil type? Or is it the habitat itself? And secondly, we want to know the origins of Erysimum menziesii. We want to know who is the closest relative to Erysimum menziesii, Menzies Wallflower. Is it Erysimum capitaneum, the western wallflower, with that crazy broad range? Or is it the more adjacent Erysimum cancinum, the Helen wallflower, often found on the periphery of the menziesii population? We don't know. And how did it speciate? What pushed it to be what it is today? And based on this, we have two current hypotheses. First hypothesis is Erysimum capitatum is the progenitor species, meaning the parent species, to a clade, which essentially is a group that is comprised of Erysimum menziesii and Erysimum cancinum. Phylogenetic trees, I know that's not anyone's bread and butter, so let me try to break this down. So that big orange triangle, imagine that represents 100 plants. And then that branch protruding outward, that represents a section of those plants diverging away from what has been stereotypically classified as capitatum, with then and as with that divergent group then forming two separate groups known as Erysimum cancinum and Erysimum menziesii. So in this instance, cancinum and menziesii would be direct descendants of capitatum. That would be the parent population to or parent to Erysimum cancinum and Erysimum menziesii. Next, our second hypothesis is Erysimum cancinum is the progenitor to Erysimum menziesii, and this is Erysimum cancinum is the progenitor to Erysimum menziesii, and this clade is sister to Erysimum capitatum. So in this instance, capitatum is on its own. It is kind of mirroring where we have cancinum and menziesii. So this is assuming that capitatum is still closely related, but cancinum in its instance would have that branch that is diverging, forming the group of what we had designated as menziesii. So menziesii would be the product of cancinum diverging. And to really approach this, well, we got to get into the bread and butter of the study, right? We got to get into the methods. We did some very thorough sampling throughout the study. We collected 223 samples from Dunn County all the way to Monterey. Each population, we collected a voucher specimen in the form of a pressing a plant and putting it into the herbarium. We also did soil sampling at each locality. I was told I needed 3/4 of a gallon bag at each locality. I only need 50 grams. So I was lugging around a lot of sand in my backpack for no reason, but now I have a lot of sand. If you want to see that, if you want to see sorry, there's mic here too. If you want to see the diversity of sand I collected, here are some falcon tubes with all of the sand I collected. Not all the sand, but right here is like 2% of that sand. So please take a gander at the gradients of cool sand I found. Someone please appreciate it. I worked hard to carry this home. Yeah, so starting in Donner County, our first site that we visited, we had two localities which cancinum could be found, or cancinum in quotes if we want to make sure to do some distinction at this point. The Tolowa Dunes Lake Earl area versus Whalers Island. There's some cancinum for the Tolowa Dunes can be found throughout it is more so widely dispersed throughout the Lake Earl and Tolowa Dune system. It can be found north of the Lake Earl as well as south of the Lake Earl. There is a lot of kind of fragmented pockets of dunes within the area, and almost anytime you see some sand, there's a little wallflower trying to make an attempt to pop up. Thanks to the efforts of removal of the European beach grass done by the Tolowa stewards, they have initiated some new substrate which the Tolowa wallflower can actually succeed and come back to. So there is a nice range and multiple range versus Whalers Island, and like the name says, it's an island. There is cancinum found on that locality, but they're adjacent to one another. They're basically in a little depression on that island. Whalers Island itself used to be a natural island, but then it was integrated into the harbor itself in like the 1930s. So it was isolated for a long time. And there's no really telling of if these two were connected at one point, if cancinum itself on Whalers Island was always separated. And it doesn't help that these two populations are now separated by the entirety of Crescent City, which is kind of built between them now. So maybe they were once a continuous population, but we don't know. Now moving south of Dunn County, we get into Humboldt County. Humboldt County, we have that Humboldt Bay wallflower, Erysimum menziesii, subspecies ericensis. And then we have Erysimum cancinum. Subspecies ericensis can be found throughout Humboldt Bay. It can be found on the South Spit, North Spit, Malal Dunes, Lamprey Dunes, Elk River as well. There is a wide range of substrates. It's all sandy substrates, but a wide range of areas that can be found throughout Humboldt Bay. Versus in Mattole, it is more so just found about 2 miles south of the Mattole mouth. When me and my lab mate Eli were collecting for Erysimum cancinum, we went based off locality descriptions on herbarium specimens. I know not all of us are familiar with herbarium specimens, but essentially it's like a museum record for a plant. Someone will collect a sample in the 60s, And then they will physically write like it was 2 miles south of XYZ. Um, and with that, there's some vagueness to the actual localities where these can be found, right? For instance, if someone says it is 2 miles south of Smith River, which does that mean like the river Smith River, or like the town Smith River in Donner County? It gets a little bit vague. So me and my lab mate Eli, we're overthinkers, and we didn't know if they meant if 2 miles south of the Mattel River meant 2 miles south of the Mattel River on like the cliffside or at the base of the cliff. So first day was exploring the cliffside, no luck at all. Second day, we were walking around more so on the Lost Coast side of it all. And after 2 or 3 hours, we were ready to give up. But then we saw a cool little seed. We were like, oh, there's probably some cool like aranthes, some monkey flowers over there. So we start to kind of go over that floor dune. Out of nowhere, we see a little head of a wallflower kind of looking right at us. So we lucked out there for sure. But yeah, that was an endeavor. So yeah, if anyone ever collects herbarium specimens, please do GPS coordinates. I would really appreciate it, and I guarantee people in the future would appreciate it as well. And with that said, I should also want to make sure to thank two people that were critical to my fieldwork before we can go any further. On the left, we have Laurel Goldsmith. She was a botanist for the U.S. Fish and Wildlife Services. She was the lead for Erysimum menziesii as a whole, menziesii. So she was maintaining the population up here. And without her, this study probably wouldn't have happened. She drove me to almost every single field site that involved Erysimum menziesii. She drove me to Monterey twice. She had to deal with me yapping for 8 hours both ways twice. Yeah, I'm a yapper. I'm sorry, y'all. Okay, sorry for her. She had to deal with that. But yeah, she carried a lot of weight this project, and without her, we wouldn't be here today discussing this project. And then when Laurel wasn't there, Eli was there. Eli went to multiple populations of Concealum with me. He was a senior in my lab at this point. So he was on his way out, but I would consider him more of a mentor. He taught me DNA extractions. He gave me a lot of grades when it came to collecting plants. At the Matol region, I said I was going to collect 12 plants. And for some reason, he thought I said 20 plants, and he was game to sit there and collect 20 plants for the next 7 hours. We only collected 12, so it was only like 4 hours, 5 hours at that point, but he was ready to roll with it. And then when neither of these individuals were there, my dog Dobby was there. So there was my emotional weight carried by her. All right, now moving on to Mendocino County. In Mendocino County, we have Erysimum cancinum as well as Erysimum menziesii, subspecies menziesii. Cancinum being the Helen wallflower, and menziesii being Menzies Wallflower. North of Fort Bragg, around 10 Mile Beach is where you can find a lot of the Erysimum menziesii, subspecies menziesii. And then kind of near Glass Beach and Pudding Creek on that coastal cliff is where you can find Cancinum. And then moving south, following that main road, you'll be able to see scattered wallflowers throughout the cliffside. There was a lot more wallflowers I wish I could collect of some Cancinum going south of Mendocino area. But when I say cliffside, I mean it was like on the face of the cliff, like 30 feet down. And extreme body's cool. I wasn't ready for it. But maybe some other day I'll be gonna know about that. But yeah, we collected a lot of Cancinum and menziesii from this locality. Moving on to Point Reyes. Point Reyes is only comprised of Cancinum, but they occur in two different areas on two different substrates. The Cancinum on the left here is found at Chimney Rock. That is found in the more stereotypical habitats, the coastal cliff, versus what is found at Abbott's Lake, which is more so a sandy substrate. So on this map here, what is blue represents Abbott Lake versus what is kind of red polygon represents where Chimney Rock is. Abbott Lake, there was a lot of individuals of Cancinum found directly on this end, but a lot of them are also found kind of in this cluster of other vegetative plants, which I'm assuming probably helps with soil retention and helps their chances of survivability within a dune system. Then after Point Reyes, we moved to Monterey. So then we get to Monterey County, where we have some of the coolest menziesii I found throughout the trips. On the left, we have Erysimum menziesii subspecies yadani. On the left, we have subspecies menziesii. Erysimum menziesii subspecies yadani, that's Yadon's wallflower. On the right, which is subspecies menziesii, that is again this Menzies wallflower. And it's really hard to encapsulate the size difference between these two. The one on the left, I would argue it's as broad as my shoulders. It forms like almost like a mat, and they all go down to one root. So there's multiple basal rosettes, but they all go to one singular like woody calyx. And then in the back, behind where you see the yellow flowers, there's some like twig-like structures. Those twig-like structures are last year's growth. This species is perennial. And I believe the only perennial Erysimum menziesii that we have within its system. And then subspecies menziesii is the tiniest wallflower I found. I would argue it's probably the size of like a clenched fist. It was tiny. The leaves themselves, they still have like that spatula shape that you typically see, where like the distal tip is kind of wider than the actual like the more proximal portion of the leaf. But the leaves were also like the size of my thumbnail. They were insanely tiny, so it's interesting to see the diversity between these two localities. So species Yadani is found throughout a more of a coastal dune system, usually behind the foredune and sometimes near some blowouts. But subspecies Menziesii was more found on a coastal cliff, but on a sandy substrate. And then you're also now seeing there is now another orange polygon, which is not associated to any of this too. Well, that was a surprise to you guys, I'm sure, and it was a surprise to me and Laurel while we were collecting Erysimum. We were told where you see that orange polygon, there was an enormous population of Ursum yandani. We went out there, we hiked for 3 or 4 hours, and all we saw was this fellow right here. All we saw was Ursum amorphyllum. So we were out there. We spent the evening assessing those populations, and we said, might as well we should collect it, right? We were out there. We should maybe this is relevant. I thought this would be a great specimen to kind of be a way to train my undergrads how to do DNA extractions. Because it's plain full on leaves, right? Lots of material. But it is a lot more relevant than we think, and we'll get into that in a little bit, a little foreshadowing. And lastly, my collection of Ursum capitaneum. Capitaneum, that is the Western wallflower, usually known for its bright orange flowers. I made a mistake of waiting into midsummer to collect this. I collected this around 4th of July weekend, 2024, and there was an insane heat wave during that time. It ranged from 110 to 120 when I was out there. So I had to plan my day around that. So, collect in the morning, hide away in midday, and then collect at night if I could. So I started up in Donner County, worked my way up to Cave Junction, collected in a dried creek bed there, went up to Mount Ashland area, collected near Mount Ashland, drove down towards Mount Shasta, and I could not find any of Mount Shasta for the longest time until I went to a naturalist and saw a post from one of my colleagues, Emily Jackson. And thankfully, that led me to a nice positive ID. Also, Emily Jackson just defended her master's thesis yesterday, so she did an amazing job. Can we give her a round of applause really quick? But also, thank you for like the 3 specimens I got from Mount Shasta. Without you, Mount Shasta would not be represented here. I then tried to collect near Chico area. All of that tissue was burnt to a crisp and not usable, so that is not in this study. I then went to Sacramento and went east towards Yosemite Valley and found some luscious Erysimum in that area. And I was fortunate enough for that. I then took my undergrads to Tail Hill to kind of demonstrate the kind of fieldwork I did, and we found some nice capitaneum blooming there. And then my advisor collected some Erysimum capitaneum on his outside the boundaries of the Lassics. So that is my fieldwork concluded, and now we get into the DNA extractions. I know you all want to hear all the nuances of DNA extraction about, right? It's a very tedious process. Don't worry, I'll keep it like 3 minutes. So in general, we extracted DNA using a kit specifically made for plants, and we sent off 190 samples to Florogenics, a company which is familiar with the sequencing we use, which is dd-RADseq. And I just want to highlight 2 individuals that were critical for my lab work, Kiana and Charlie. Kiana got funding through my advisor, Dr. Oscar Vargas' NSF grant, and Charlie got the first in family grant. They were both paid to assist me so they could fully embed their teeth into this project. So it's cool to have 2 individuals gung-ho and be able to financially support themselves to invest themselves emotionally into this project. They assisted me with over 300 extractions. We also got to do this cool little technique with liquid nitrogen. And essentially, anyone has cooked an egg here, right? When you cook an egg, you're denaturing protein. That's because it gets hot. With DNA, you don't want DNA to get hot because then it becomes degraded. So lowering the temperature gave us more of a buffer. So liquid nitrogen was our savior for DNA extraction, so we'd get good quality DNA. And then from that, we set up our best 190 samples to Florogenics. And then lastly, my phylogenetic methods, and similar to DNA extraction, don't worry, I'm literally just mentioning these two bullet points right here. I use a pipeline that was specifically made for DD-RAD sequencing. And then with the output of that, I threw it into a program called IQ Tree, and we got some cool results. So phylogenetic trees are always hard to comprehend, especially during a talk, right? There's a lot going on here. I mean, I spent the last year looking at this, and I still keep noticing small nuances. So this is a collapsed tree, meaning those tips represent usually multiple individuals. The colors associated to what taxa they're associated with. And before we dive deeper, I kind of want to go back to my initial hypotheses. The first hypothesis was that Erysimum capitaneum was the progenitor to the group that defines Erysimum cancinum and Erysimum menziesii. Right away, you look at this phylogenetic tree, you'll notice about 3/4 of the way down. Sorry, I think I can move the cursor. Right here. That is all of the capitaneum that I collected. There's roughly 13 specimens there, I believe, and they all formed their own group. We do not see like cancinum and menziesii branching off of that cluster at all. So right away, my first initial hypothesis, well, it's a no-go, which is I think it speaks to the complexities of this project. Then falling forth with my next one, we don't see capitaneum. We do see a adjacent cancinum to. Sorry, I'm trying to move my cursor over here. We do see an adjacent cancinum to the capitaneum population, but then we don't see a cluster of cancinum coming from that. We don't see a cluster of menziesii coming from that species. We don't see it diverging from that cancinum population. So my second hypothesis right away is that's a no-go, which is not bad. I think it speaks to the complexity of what's going on here. So I want to highlight there is 4 separate groups where we see that those yellow tips form. Essentially, this means there is 4 different potential species of Erysimum menziesii. These all converge on their own. They all came to a same set of outcome depending on their environments, and the speech my worst portion. They all, by chance, look similar to one another. You'll notice in Mendocino County and Monterey County, you'll notice that there is cancinum kind of surrounding the menziesii in that Mendocino area. So this cluster of menziesii is surrounded by this group of cancinum. This suggests that this population of menziesii is derived from cancinum in Mendocino County. Now I'm jumping down to Monterey. Going back to what I mentioned earlier, Ammophilum. We have Ammophilum right here, and then branching from Ammophilum, we have menziesii. So Ammophilum, the accidental collection, is the progenitor to the menziesii population within the area. So that was a nice wild card, and it was pretty cool to see. You'll also notice that there was 4 separate groups of cancinum as well. So Erysimum's complex. We have 4 separate origins of cancinum, or at least what we define as cancinum occurring here, which suggests the potential for 4 different species of cancinum occurring. So yeah, potentially 2 species become 8 species, potentially. And what is driving this development? Well, why are we seeing what is driving this? What is the force pushing it along? Well, it's probably the coastal dunes itself. The coastal dunes is probably the proxy for convergent evolution in this environment for this group. Coastal dunes, they're harsh, right? They're full of how many of you hiked through the dunes, got blasted by sand? I'm sure most of you, right? Then you throw in some salt spray from the waves crashing. That's corrosive. That is a tough environment. Not every plant can make it there. So it would make sense that a particular set of traits would enable those plants to succeed. It's also full of it's also known for having low nutrients. There's not a lot of moisture in sand. Sand is mobile, it is abrasive, and it is suffocating. I.e., sand can move and pile up on top of a plant and kind of choke it out from existence. So sand is probably a critical factor with these distinct menziesii populations we are seeing. And also, Erysimum menziesii is a critic species. Critic species refers to a species that is near identical from each population, but genetically they differ from one another. Critic species are notorious for happening in areas with rapid divergence, such as we see in the California Thurston Province. The genera Erysimum as a whole, the wallflowers, is notorious for undergoing rapid divergence as well. So we have an area prone to rapid divergence, i.e., plants, forming in their own evolutionary direction, And then we have a plant that's also renowned for it too. I think it was just the perfect storm for diversification. So in conclusion, Erysimum menziesii seems to be more so of an ecological species. And there's potentially four unique species. The Tolowa wallflower is sister to Weathers Island Erysimum cancinum. They're not the same species. Humboldt Erysimum menziesii is closely related to the wallflowers found in Donner County. Mendocino Erysimum menziesii is derived from Mendocino, or that's supposed to be Erysimum cancinum. Sorry, that's my one mistake today. Thank you for laughing at that one. I appreciate it. The Monterey Erysimum menziesii is derived from Erysimum amorphyllum. And we have convergent evolution in a cryptic species. Erysimum cancinum as well, the Halen wallflower is not a natural group. A natural group would mean all these would form one group on a phylogenetic tree. We have four separate groups, so that's just that these are likely four different species as well. And why is this taxonomically relevant? Well, the Tolowa wallflower should belong with the ecological species of Erysimum menziesii. It deserves that 1B point. Sorry, that's a typo. That's my second mistake. 1B.1 CPS rank that Erysimum menziesii is currently attributed. And sad also is just something that's kind of dire, I would say. We cannot just replace one population with another. We cannot just replace Dumble Bay wallflower with wallflowers from Mendocino County or Monterey or Donner County. All these are unique lineages. If one population gets wiped out by, like, let's say the white rust that's found locally here, we can't really bring it back to that. So we probably should make sure to build a pretty solid seed bank just in case, right? A lot of these areas are known for anthropogenic disturbance, right? Invasive species. This species, which is already endangered and critical, is even more, I guess, critical and endangered than we previously perceived, which is like cool results, but there is some weight with that. And now for the fun part. Woo, acknowledgements. Sorry, this is a wall of text here. I will try my best to go through it very quickly. For fieldwork, of course, I want to thank Laura Goldsmith and Eli Allen. Eli and Allen also assisted with my lab work, as well as Kiana and Charlie. Susan Wright is the one that helped me with the look and action plan. I appreciate her so much. Thank you to my committee members, Oscar, Rachel, Eric, and Chloe, for their assistance with this project coming to be, as well as the rare plant committee through the the local CPS chapter. We had a lot of back and forth with my proposal, which really gave me a great understanding of what's going on with Erysimum, and this project wouldn't be here without them as well. I want to thank all the members at Vargas Lab. Without you guys and your mentorship and or your companionship, grad school would have been a lot heavier. Grad school is hard, right? Life is hard. Having a core of people that are there to support you and to bounce off of you, like that community is valuable, and I will always appreciate the friendships I made throughout this time. Same thing for my cohort. Thank you all for being here today. I also want to thank the U.S. Department of Fish and Wildlife for permitting. Thank you for allowing me to collect some rare species. I also want to thank the CPS Student Research Grant, the statewide one, the CPS North Coast Chapter Student grants, the Northern California Botanist Student Research grants, the Tolowa Stewards, and Senator Beck for their contributions for this project as well. The Cal Poly Humboldt's Master Grant, the First Family Grant, and the Rumbo Award. I also want to thank Alclay Rancheria for supporting me financially throughout my time as a grad student and for giving me a job right now. That helps a lot as well. Sorry, I stole a joke from Emily. She did something yesterday. Yeah, it's nice to have financial stability for a little bit. And that concludes. Okay. Also, sorry, one thing I want to say. Thank you for all being here today. This is an amazing turnout. It's so awesome to see so many of my old students, colleagues, and friends. Thank you. Okay. So does anyone first off, does anyone have any questions? Here is my lovely Dobby. Laying right next to a population of Cancinum at Fort Bragg area. She was taking a nap while I was working away. If you don't want to ask a question right now and you want to shoot me an email, scan that QR code. That'll take you to a GitHub page that has my email, also the email to our lab at Cal Poly Humboldt. Right, email the link to that page. But yeah, does anyone have any questions? I'm gonna take a sip of water really quick. There's not Red Bull in here, I swear. Yes, Paul.
Yeah, so I'm not really ready to let go of hypothesis two. I mean, it is more complicated, but especially if you fiddled around with the routing here, which is not totally clear, it does seem like you could maybe make the bluff one be the progenitor for 4 different plates within it.
I think that was more so contingent on everything being monophyletic, and Cancinum and menziesii are not monophyletic. They're not just one solitary group, unfortunately. Any other questions? Yeah, Kyle. So Kyle's question was, what was my favorite spot to collect from? God, that's heavy because there's like interactions that had those spots which tie into it. But the one that blew my mind away was honestly the Adon's flower. Like that population is it went against everything I associated with like the local wallflower here. The leaves were thick. They had slight hairs with 4 or 5 different like rays going off of it. The leaves themselves were like more succulent like. They were such an oddball to me. And I get why Robert A. Price back in the day tried to say that was their subspecies. It is distinct. It is cool. Also, this is my first time going to Monterey through this project, so Monterey was beautiful. All the golf courses hurt to see. And I wonder if that played into the fragmenting populations, but that's a whole nother conversation for a whole nother day. That's something I know they did sand mining in Monterey area. They used to mine the there for glass, which I'm wondering if that also contributed to disturbance within East Texas. The conversation for another day. Yeah, Carol?
So I'm thinking about what I learned is that the wallflower out in the dunes is endangered.
Yeah.
So what you're saying still is it's endangered.
Mhm.
But on your maps, it doesn't look any rarer than the other populations.
You mean like by like the Madonna you see? That Oh yes. Carol's question was I'm saying that Erysimum is rare, but a lot of occurrence data on those maps. I would argue there's something called observation bias. Most of my data was from a what's it called DPIF, which is from iNaturalist. And those areas are probably just more accessible to the public to take photos. So then people can post more occurrences of them versus like the Cancinum, which is usually more remote regions, like the Lost Coast, it's a trek to get to from here versus being like 30 minutes, west, or even 30 minutes west, it's just down the road from here. So I think it's just bias of how easy it is to see those plants versus how far you drive to go see Cancinum. And a part of it, the endangered ask too, is also associated with the environment it's on, as well as the invasive species affecting it and other anthropogenic disturbances. So when they consider rare plants, from my understanding, they also will see like, all right, is there a lot of off-road activity here? Is there a lot of foot traffic? Is there a lot of home developments? That also does play into the factor of if it's endangered or not. In Monterey, there's lots of home developments. And back in the day, I'm pretty sure there was supposed to be more developments out in the new systems here, but never went to fruition. Sorry for there's a lot of factors that contribute to rare plants. And I think a lot of the excess occurrences we're seeing here is just people being more inclined to hike a nearby dune system versus driving 3 hours south of here to see a Cancinum. Kiana. I would love to. I still have a few more analyses I want to keep doing. I am trying to get into PhD programs. We'll see if that can align to whatever I'm doing in the future. If not, maybe one day when I'm a professor, I can bestow that upon the next generation. Because Kingdom is so cool. There's more populations I wish I could address. There's a few populations on rocks, kind of in coves, which would take something called extreme botany to occur to get that. A little bit of kayaking, a little bit of rock climbing. I was too chicken, unfortunately. I also needed insurance, which I didn't have. Insurance is expensive. But you never know. Maybe that could be a part of future study. One thing I would like to know myself is, was there some kind of pre-adaptation that occurred with Erysimum? Did something in its historic past kind of contribute to all them having a similar trait at the end of the day? Did they go through another like endemic environment, meaning a harsh environment? So like the dunes is a hard environment. Maybe the Western wallflower moved through like certain soils and had to adapt some resilience from that, and it kept moving westward until maybe if the Western wallflower is so closely related, maybe there's some gene flow that perhaps it picked up a trait from an ancestor in the past. So that'd be cool to figure out. Until then, I'm just gonna keep pondering on that every night. Yeah, Paul.
So I think maybe if people don't want to see the phylogeny anymore, we could go to chat. And see what's happening on Zoom. We got a text.
Sorry, I'm being a boomer.
We might need
Oh no.
Yeah,
To see if people had questions, is that?
Well, I got a text saying. Something. You can talk with the audience while I'm messing around with this. if you
Actually,
want.
Yeah. Yes. Hand right there.
That's compared to most basil in the students.
And repeat it.
So the question was, is there a genus that's considered most basal, or sorry, a species that's considered most basal with this genus? There was a paper in like 2018 where it was addressing Erysimum as a whole. Erysimum as a whole can be found throughout most of the northern hemisphere, in China, Europe, etc. From my understanding, the North American lineage is newer. It is younger than the rest. But I'm pretty sure Capitame is probably one of the Capitame is considered one of the older species within the genus in North America. I need to go down more than rabbit hole of the European lineages, but that's a whole other battle. There's also between 150 species or 300 species, depending on the author, which alludes to how vague the populations all can be.
Yeah, I think read the question slowly for this audience and then answer it.
Perfect. So Susie had a question. I wonder if this genetic work will help reestablish the subspecies of the Humboldt Bay wallflower. My understanding is it is not currently an official subspecies due to the lack of genetic support. That is correct. All those subspecies I mentioned, they were proposed by Robert A. Price, but they never successfully were established due to the lack of genetic information at the time. If anything, I think this more so supports that these might be a new species. So perhaps one day, potentially, Erysimum, the Humboldt Bay wallflower will be Erysimum ericanthus rather than Erysimum menziesii subspecies ericanthus. So it definitely supports its subspecies at least, but more likely supports that this is a whole new species. Depending on some more other analyses, but my structure analyses, which is a way to see kind of genetic clustering, as well as my PCAs is supporting that so far. Like there's like a lack of gene flow. And I think, is there more if I gotta scroll up? Great talk.
So you try to name.
If I have the opportunity to name them, I don't think we are not from this area, right? Like, I'm sorry, this is gonna be a small tangent. I guarantee I was not the first person to see these wallflowers, right? There is indigenous communities in this area. I feel like they have more of a right to name these species than we do. So if there is a chance we can name them, and if someone in the past didn't try to name them already, I will likely talk to the local tribes of the area and talk to them about naming these species. It's not my right to take that name.
I mean, they're gonna need guidance from someone who understands the botanical.
Yeah,
process.
so I'll guide them through the nuances if I have to. Yeah, and like with me being Tolowa, I would love to give that to like the Tolowa Dani as well as Uncle Evangelia to discuss that name especially for the donor population if possible.
Chad.
I think that appears to be it from my understanding. It's just rather perfect. Well,
People can use your QR code, so
Yeah.
thank you so much.
thank you all for being here. Thank you for everyone being online. Yeah. And yes, please feel free to use this QR code if you have any further questions. Also, I'll be doing my dissertation next semester. My thesis defense. Sorry to use my words. I'm a little bit tired. If you want me to email you that date, whenever that occurs, please shoot me an email and I will put you on the email listing for next semester. And yeah, thank you again for being here. I appreciate you all.
Before you all go, I forgot something, one of my announcements before we started. I was so anxious to introduce our speakers. Is this not working? Nobody's listening. Okay. Okay.
Look at this.
If anybody is listening, maybe if people on Zoom are listening, we need someone to help deliver flyers for our upcoming spring plant sale. We need someone to go to the College of the Redwoods, someone to go to Mad River Gardens, Humboldt Botanical Gardens, Humboldt Bay National Wildlife Refuge, and then the towns of Rio Del, Carlotta, Lolita, and Ferndale. Again, talk to Karen at the merchandise table if you would be willing to help with any of these distributions for help advertise our plant sale. Thank you all for being here.